Okay, so our speaker tonight, I'm very pleased to have Dr. Jane Watson on board to talk about sea otters. Jane got her BSc at UBC and PhD at the University of California in Santa Cruz. And she's now the emeritus professor at Vancouver Island University, where she taught for 20 years, I think, plus. So, and I know for my mind, I associate Jane Watson with sea otters. Whenever I heard sea otters, I think of Jane Watson. So I'm sure she will have a very interesting talk. So with that, I'll hand it over to Jane.
Okay, thank you very much, Phil. This is my first full-length presentation, so it's really odd. I'm used to pacing about, waving at people and doing silly things, so we'll see how it goes. What I'm actually going to talk about is I love to lure people in. They think they're going to hear about warm, fuzzy sea otters, and in actual fact, sea otters are just a medium for me to look at kelp forests. So you're going to expect to hear about warm, fuzzy creatures, and instead you're going to get a lot of cold, slimy kelp. But I do need to start off by explaining why sea otters are so important. And this is one of my favorite quotes. It was from a French naval captain, La Perouse, whose whose La Perouse Bank is named after. He was a naval captain who sort of followed in the footsteps of Cook in terms of his approaches. He was into scientific discovery and exploration. And in his trip along the west coast of what is now British Columbia in 1786, he noted that sea otters were more common than seals on the east coast of North America. And I don't know if anyone has seen how many seals are on the east coast of North America, but it's really quite remarkable. The interesting thing is to note that but 24 years later, Captain Ebbets, writing to his boss, John Astor, who was a fur trader or fur exchange trader in New York, he comments that the fur on the northwest coast this year have been very scarce. So this points to what happened. We went from something that was incredibly common and abundant to a species that was extraordinarily rare. And this came about largely because of a large-scale commercial fur hunt. When Europeans arrived on the coast of what we now call North America, they encountered a culture that was very well versed in both capturing and in trading in sea otter furs. Sea otter furs were worn largely as regalia by high-ranking members of First Nations cultures up and down the coast. But with the arrival of Europeans, a large-scale maritime fur trade began. It started in the late 1900s, and no one knows exactly how many sea otters there were in British Columbia, but at least 55,000 pelts were traded and landed. At the height of the sea otter fur trade from 1799 to 1801, about 12,000 pelts a year were traded. So these truly were abundant animals. Sea otters were probably rendered ecologically and almost economically extinct by the mid-1800s. The problem was they had become so valuable that even though they were extremely rare, hunting for sea otters was very, very well worth it for people. So as the animals became more valuable, hunting pressure increased. In 1911, sea otters were finally protected under terms of the International Fur Seal Treaty. There were probably fewer than 2,000 sea otters left throughout the North Pacific. And by 1929, the last 3 sea otters that we know of in British Columbia were shot. Now, this turns out it was not to be the end of the sea otter in British Columbia, because from 1969 to 1972, 89 sea otters were introduced to this location here, Checklisset Bay on the west coast of Vancouver Island. And these animals were brought from 3 locations in Alaska, initially from Amchitka Island in an attempt to mitigate the effects of nuclear testing that was being done on Amchitka Island. This is the site of the location in the Bunsby Islands. It was a beautiful area, lots of shallow rocky reefs. It was deemed by Carl Kenyon, the expert at the time, to be ideal habitat. So from 1969 to 1972, 89 sea otters were released into Checklisset Bay. It was probably mostly the last sea otter translocation of 42 animals that gave rise to most of the sea otters we have now. The first true translocations were fraught with difficulties. And it was in the third translocation when they actually held them in net pens and only released them as individuals were deemed fit for release. So sea otters have done extremely well. From the 89 animals that were released, the first count was done in 1977. They counted 77 animals. The most recent count was done in 2019. And at that time, we counted about 8,000 individuals, a half of which would be mature. So this is all individuals in the population. This is a pretty rapid growth rate. It averages about 8% over that entire time span. And it's pretty hard to imagine how a population could go from 89 introduced animals to 8,000 so quickly. But the fact of the matter is that sea otters, when reintroduced to areas, do well because they have abundant food supply. The females, although they bear only one young, are very good at looking after their offspring. So they have a very high success in weaning their offspring. And other than humans, they have few natural predators along the west coast of British Columbia. So in addition to growing very rapidly, the sea otters have also spread. They were introduced here to Checklisset Bay. They now extend as far south, essentially, as they've been seen as far south in large numbers of Amphitrite Point, but they move back and forth, and they occur all the way north to Aristazabal Island. So a very wide range. I just want to point out that I've just given you a whole pile of very impressive numbers. And I want to point out that these data are trend data. They're numbers that are generated by people counting otters from small boats. There we are counting otters. And I want to point out that to really get an idea of what we're doing, there's a raft of otters. So I think if everyone studies it, they could probably count the otters. But what I want you to do as you're counting them is move up and down and back and forth and try and come up with an estimate of abundance. So these numbers are an estimate, and we hope that the counts are collected consistently each year so that we're getting a fairly reliable growth trend. I know I don't need to do this to a natural history society, but I do it every time anyway. This talk is going to be mostly about sea otters, which have a very close relative called the river otter, which in our part of the world lives in the marine environment. And I'm afraid I regularly get emails saying that somebody has got a family of sea otters living under their house. I am welcome to come and get them if I would like them. River otters are quite different from sea otters. They're much smaller. They're about 13 kilograms in size. They're highly terrestrial. They're very comfortable and adept at moving on land, and of course occur all across North America. River otters den on land, and they give birth to up to 4 offspring. River otters have webbed feet to swim with. Sea otters are much larger. A male sea otter can grow up to 40 kilograms. That's the size of a German Shepherd dog. Like all marine mammals, sea otters give birth to a single young, which they tend to for about 8 months. Unlike river otters, sea otters have flipper-shaped hind limbs, which they use for propelling themselves through. This is where I need to be able to pace around, propelling themselves by lying on their back and using their flippers. Sea otters haul out on land occasionally, usually restricting themselves to the intertidal zone because those flipper-shaped hind feet mean that they're really not very good at moving about on land. Sea otters are also animals that feed on invertebrates. And unlike all other marine mammals, which depend largely on blubber to keep themselves warm in chilly North Pacific waters, sea otters instead depend on a really thick fur. It's about 200,000 hairs per square centimeter. And the hair is composed of long guard hairs on the outside and short fuzzy hairs on the inside. And they're constantly grooming and blowing air into that fur. The guard hairs trap the air against the body, and then the body heat warms that air up and provides insulation. So along with this really thick fur, sea otters have extraordinarily high metabolic rates, about 2.5 times that of a similar sized terrestrial animal, which doesn't really mean very much to most people until I tell them that a sea otter has to eat about 25% of its body weight in food each day. And even the most hungry teenage boy is hard pressed to eat 2% of his weight. So this is a great deal of eating. And it turns out it's what makes sea otters so important. Sea otters are probably best known for eating urchins, and that's partly an accident of nature, which we'll come to in a minute. These are red urchins that have been eaten by otters. They flip them over and they go in through the oral surface where the spines are a lot shorter. It makes it much easier to eat an urchin. The important thing about urchins is that they are grazers. I tend to call them the lawnmowers of the sea. And urchins, through their grazing, can control the abundance of seaweed and also its distribution. So in British Columbia, seaweed is generally in areas where there's lots of urchins. Seaweed is restricted to shallow subtidal fringes where wave action prohibits urchins from moving into shallow water, or algae will grow on unstable habitat where rocks and such roll about so that the urchins can't graze. So in areas where there are no sea otters, we end up with systems that are often referred to as urchin barrens. And these are systems that are dominated by urchins and have very little fleshy algae. All this pink stuff you see is coralline algae. It's seaweed that is encased inside a calcium carbonate skeleton that prevents the urchins from grazing it. And these densities of urchins can get extremely high. These are purple urchins in Houston Stewart Channel in Guayhanas, a picture taken by Lynn Lee, but you get the impression of how dense these urchins can form. Of course, as I've said, sea otters eat urchins. And as you can well imagine, if you take the grazer out of a system, seaweed starts to grow. And so as otters move into rocky subtidal areas and remove the urchins, kelp generally replaces the sea urchins. And so you can think of this as alternate stable states, if you like, two systems that are both very, very stable. We have an urchin barrens in which you have lots and lots of urchins and very little fleshy algae. When you add a sea otter to the system flips over and becomes a kelp forest. And what I'm mostly going to talk about this evening, remember I said I was going to lure you in and talk about kelp, is I'm going to talk about some of the things around these changes and some of the long-term shifts that we're seeing. However, I am a bit of a proselytizer when it comes to kelp, so I need to make sure that having told you why otters matter, I'm going to tell you why kelp forests matter. So the first thing to understand is that kelp forest increases nearshore productivity, and it does this through a number of means. One is the kelp sort of breaks down into these tiny little particles, and those tiny little particles in turn fuel detritus-based food webs. Kelp leaks carbon into the water, and that carbon in turn is consumed by organisms. And that means that these organisms have a greater growth rate. So they can grow 2 to 3 times faster in a kelp forest than in a comparable urchin barrens. And this has been tracked by looking at the isotope of carbon that gets incorporated into animals. Kelp incorporates the isotope carbon-13, and most other photosynthetic organisms are incorporating carbon-12. So you can track this growth. So increases productivity. The next thing it does, of course, is it increases nearshore habitat, particularly for things like fish. And this is a school of juvenile young of the year boccaccio, which are rockfish species that are listed. And this is in a kelp forest in Checklisset Bay where sea otters were introduced. So in general, Russ Markel and a number of other authors have found that wherever you get lots of kelp, you get far more fish. In turn, this kelp provides surface for all sorts of other small organisms, which in turn provide food for larger organisms. So kelp increases habitat. And then something that most people don't think about is kelp also changes the hydrodynamics of an area. And I'm not going to talk too much about that tonight, but it can be very, very important. As kelp grows up from the bottom up to the surface, what it does is it slows water as it moves through that kelp forest. And in doing so, it means that it's going to trap the larvae that are released from invertebrates within the kelp forest. So it's going to entrain things, it's going to retain them, and it's going to completely change patterns of recruitment. So you can see these huge encrusting fauna that are growing up beneath this kelp forest. Likewise, those kelp forests reduce onshore erosion. They act as natural barriers to reduce erosion and sediment transport. Okay, so that's why kelp matters. Now, this whole story about sea otters, sea urchins, and kelp was a story that was first told by the man that I did my PhD with, a man named Jim Estes. And he described different nearshore community structure along Aleutian islands. He found that at islands that had sea otters in the Aleutians, there was lots and lots of kelp. And at islands without sea otters, there was virtually no kelp and lots of urchins. So this sort of idea that you could have alternate stable states was described for Alaska, and then also by a man named Jim McLean, in California. 35 years ago, when I started my PhD, no one had actually documented the actual change from an urchin barrens to a kelp forest. My good friend, Paul Breen, along with a number of his colleagues, had compared areas with and without sea otters in British Columbia and shown that the paradigm seemed to be true. Areas with otters had lots of kelp and no urchins, whereas areas without otters had lots of urchins and no kelp. So I set out 35 years ago to try and actually track the effects that sea otters had on nearshore structure. The problem with tracking changes is what you really want to do is look at how things change over time. And it's very easy when you're sampling underwater to accidentally think you've gone to the same location, but in actual fact, what you're sampling is an area 10 or 20 meters to the left or right of where you sampled last year. So to try and track changes over time, I set up a series of permanently marked plots. And these plots were set up, 2 of them in Checklisset Bay, the red arrows here, 2 of them in Barkley Sound here. The 2 in Checklisset Bay were set up in areas where there were sea otters, and they were designed to look at how variable systems that already had sea otters in them were. And then I set up 2 sites in Barkley Sound, and these were designed to examine how variable sites without sea otters were. And then there were 4 sites scattered along the west coast of Vancouver Island. And these were sites that I hoped sea otters would invade, and I would be able to watch the transition from urchin barrens to kelp forests. I always like to point out that no one in their right mind would do this for their PhD, but I got lucky. So each one of these permanently marked sites is 25 meters long. It has stainless steel pins set in the bottom. And once a year, we go down, we find these little, I don't know if you can see it here, there's a little flag that's on a stainless steel pin. We find these stainless steel pins and we run a tape measure out along each of these 5 swaths. So 1, 2, 3, 4, 5. And then divers go down and they swim along the tape measure, counting all of the algae and invertebrates 1 meter on either side of each of these 5 swaths, which gives us 5 10 by 2 meter swaths. And here's Leah's. This is last Thursday, actually, at Wizard Island, and you can see the tape along the bottom there. That's the swath. Here's a 1 meter counting bar, and there Leah's busy counting all of these urchins and sea cucumbers that are at this urchin-dominated site. So what I want to do is talk a little bit about the changes that occur in kelp over time. And I just need to tell you what seaweeds we're going to talk about. The first are 2 species that are annual. The first is Desmorastio Ligulata. I'm going to call it acid weed. And acid weed's a really cool plant. Here it is right here. It actually has sulfuric acid in cells in its epidermis. And that acid is designed to deter urchins from grazing it. Bull kelp, you all know what bull kelp is. It's also an annual. It's the fastest growing photosynthetic organism. And it forms a canopy in about 10 meters of water, but it usually gets ripped up by winter storms. The other canopy forming kelp is Macrocystus, and Macrocystus is also known as giant kelp. It is a perennial, so it lives certainly up to 5 years, and it forms huge canopies at the surface in about 30 feet of water. And then, oops, thought I changed that. That should say stalked, not stalked. Stalked kelps are perennials, and these are kelps that live anywhere from 13 to 20 years. They form a canopy that of about 2 meters high off the bottom of the seafloor. And there's 3 species, Pterygophora, Laminaria, and Icinia. I'm going to talk about Pterygophora and Icinia. Okay, so we're going to start off by looking at one of those sites that doesn't have sea otters at it, Wizard Island, which you've already seen. And I always use this slide just to point out that these sea urchins are looking for carbon. And a tape measure is made of carbon. The life expectancy of a tape measure on the seafloor is about, 45 minutes. So you have to work fairly quickly. And that's what this urchin here is doing. It's eating my tape measure. So all of these slides are going to be exactly the same. Most of you probably do your own income tax. I do not do mine. So these are really simple slides. What we have across the bottom is year 1987 through to, this is data that's hot off the press from last Thursday, 2021. And what I've got on this axis, the y-axis, is the average number of either urchins or brown algae per 20 meter swath. So each one of these dots is an average, and then these little bars are called standard errors. Forgive me for all of you that already know this, but the point is that these error bars are measuring how variable the count is across all 5 of those swaths. So when you see a really big bar like this, it means it was highly variable. What I want you to notice about this graph is a very simple take home. Urchin abundance has remained relatively constant, and there's very, very little seaweed. So in the absence of sea otters, urchin barrens remain as a fairly stable, predictable system. So now we need to go into sites that have been continuously with sea otters throughout the time I've been sampling them. And you'll remember that these are going to be sites that have lots and lots of seaweed. And so this is Gull Island, which is located very, very close to where sea otters were reintroduced. So it's probably had sea otters at it since about 1975. And I want you to notice the scale here. It goes all the way up to 1,200 per 20 meters squared. So what I would like you to notice about this is, first off, there's no urchins. The otters have taken care of them. You have a few urchins over here later on, and we'll talk about that in a little bit. But what I want you to notice is that up until 1997, the abundance of kelp at this site was fairly consistent. And then something happened in 1997, absolute chaos. And then we get a slow decline in this chaos and back to this nice, regular, predictable system. So what I want to do is take that brown algae and divide it into the annual and perennial species. And what you'll notice is almost all of that chaos is brought about by changes in the abundance of annual algae. And look at how tremendously variable, these huge error bars. But if you take a look, you'll notice that the perennial algae undergoes a slow decline. It's at fairly low abundance while all that horrible annual algae is there, and that it undergoes a bit of an increase and stays steady. So if you take a look, this perennial algae is mostly that pterygophora, that fairly long-lived perennial species. We're going to come back to the story about what's going on here. The annual algae is mostly acid weed. And what literally happened was in about 1998, all of the pterygophora disappeared from this site. It created open space, and we got this masses and masses of Desmorastia. And I won't tell you how hard it is to find a little tiny yellow flag under 1.5 meters of waving acid weed, but it's not very pleasant. But you'll notice what has happened is by about 2016, the acid weed had pretty much disappeared, and the site was dominated once again by pterygophora. And we're going to come back to that story. Just very quickly to look at the sites that sea otters invaded, the best sign I ever had was the presence of these otterized urchin tests. And so here's Union Island, and we set this site up in 1988. It was an urchin barrens. There was 100 to 150 urchins per swath. In the spring of 1990, 120 male otters set up camp over top of this permanent site. And when we went and sampled it in 1990, there were no urchins, and it was a sea of seaweed. The seaweed declined over time and has sort of undergone these fluctuations, but you'll notice that there are virtually no urchins, except maybe for a tiny little bit of an increase here in 2017, 18, and 19. So we're going to divide this again into annual and perennials. And so at this site, the otters came in the middle of summer. They arrived, they removed all of the urchins, and the species of algae that recruited was bull kelp. Bull kelp is fertile in the summer. All of a sudden, there's no grazers. It went gangbusters. But over time, that bull kelp was replaced by perennial species, in this case, a mix of Macrocystus and pterygophora. And you can see some fluctuations over time. Mostly these are declines in pterygophora. But you can see that pretty much the site, once urchins were removed, has remained algal dominated. So that's the pattern I expected: otters coming in, eating all the urchins, and lots of kelp. But that's actually not what happened at 2 of the sites. And the example I'm going to give you is Cayucit Bay. The site was set up in 1987. There was about 275 urchins per swath. It was a fantastic urchin barrens. There was still quite a few urchins at that site, probably 225 per swath or so in 1989. But somewhat puzzling was the fact that there were areas where there were no urchins and lots of kelp. So kelp abundance had increased to about 100 plants per swath. And if you look at what's going on here, these are those huge error bars again. From 1989 right up until 1993 rather, there were lots of urchins at this site. But the odd thing was some of the swaths had urchins stacked 3 and 4 deep, whereas other swaths had absolutely no urchins and lots of kelp. And by 1994, otters had finally removed all of the urchins. There were no urchins right up until the very end here, 2019 or so. And you can see we have these fluctuations in brown algae. So it seems like at Cayucit Bay, sea otters came in and picked at their food, and somehow or other that created this very patchy system. So if you take a look at what happened in terms of the seaweed that grew, For the first 5 years at this site, for 6 years at this site, we get a real mix of both annuals and perennial algae. So you have bull kelp recruiting, and then it gets replaced by perennial algae. And then we have this horrible spike here. You're never going to guess what that is, acid weed. And then that acid weed disappears and we end up with pterogophora again. So the really cool thing about this site is sea otters came in, they picked at their food, they ate urchins. And what we noticed was that at the sites where there was, at the swaths where there was lots of seaweed, there were also urchin tests that had been eaten by otters. So it seemed as if the otters were coming in, picking at their food, eating an urchin or two, discarding the urchin sunk to the seafloor and created an escape response in the nearby urchins who looked at a dead urchin shell and went eek and ran away. So we ended up with urchins running away from these urchin tests that were on the bottom. And this created holes in the urchin barrens to which kelp could recruit. And we're going to come back to this as well, because I think it's really important. So this would be a behavioral response in the urchins if it's true. And here's an example of a behavioral response that my colleague Lynn Lee took. This is a large sunflower star, and you can see that all of the urchins are clearly avoiding the sunflower star. So what we proposed was that sea otters come in, they eat urchins, and if they're only picking at a few urchins, as they drop the urchins, those urchins fall to the floor. The living urchins flee from the dead urchin tests. They create areas of exceptionally high density of urchins, and then also areas where there are no urchins, which allowed the seaweed to recruit. So this is an idea that is fairly testable. And so we set out to try and do that. We set up a series of 5 by 5 meter quadrats on the floor, and these were all strung with string and bars so we could actually map urchins. And what we did was a series of experiments. We pretended to be otters, and we cut open the bottom of a bunch of 10 to 12 urchin tests. We scooped out the innards, deposited that somewhere else, and then introduced these 10 to 12 urchins into the middle of the quadrat. The second experiment we did was to realize that, well, maybe these aggregations of urchins have got nothing to do with the urchin tests. Maybe the urchins are all dogpiling on top of the kelp that is somehow managing to recruit to areas that otters have taken urchins from. So I created what I called kelp kebabs and put kelp on some rebar and stuck that rebar in the middle of 3 of these quadrats and then mapped what the urchins did. And then just to make sure that what we were seeing wasn't some sort of behavioral response to increased urchin density, as a control, we introduced 10 or 12 living urchins into the middle of 3 of these quadrats and looked to see what would happen. And I'm not going to show you everything, so you don't need to worry. But here's the 3 experiments. So we're going to add urchins, we're going to add kelp, and we're going to add otterized urchins. So this is one of those 5 by 5 meter quadrats. Each one of these dots is a sea urchin. And this is what this quadrat looked at when we started the experiment. And this is what it looked like 24 hours after we had added 10 to 12 perfectly healthy urchins into the middle. Nothing happens. So then we did the same thing with the kelp kebabs, which I'm not sure if you can see the top there, but this is what the quadrat looked like before we added the kelp kebabs. This is what happened 24 hours after adding the kelp kebabs. You can see there's some urchins all piling on top of the kelp. And then this is what this same quadrat looked like 96 hours later. It really, these 2 are indistinguishable. However, when you add otterized urchin tests, here we are at time 0, we add 10 to 12 urchin tests that have been otterized into the middle. This is what it looks like 24 hours later. This is what it looks like 96 hours later, and 10 days later, it looked pretty much the same. There were no urchins. So it was pretty clear that the response that we were seeing, these big patches being cleared, was probably created by these eviscerated urchins being dropped to the seafloor by foraging sea otters. And I think this really matters, and I'm going to tell you why shortly. But I just want to recap then what we now know about kelp forests. So we know that these kelps seem to be undergoing a pretty predictable successional pattern, much as you would expect in a terrestrial forest. Sea otters come into an area, they eat the urchins. And generally speaking, if the urchins are cleared in the summer months, we're going to get the species that are available to settle. It's probably going to be annual species. If they do this in the winter, it might go straight to some of those stipitate perennial kelps. And so those annual kelps will always be replaced by long-lived perennials. And so what you would predict then, if these long-lived perennials are living 10 to 20 years, you would predict that over time, these seaweed communities should generally become more and more predictable. And I've just dragged you guys through three graphs that say these kelp forests, in actual fact, are anything but predictable over time. And so what's the reason for that? Well, there's a few reasons. This is all pterygophorus stipes on a beach in the Goose Group Islands on the central coast of British Columbia. And what happens is sea otters move into an area and remove all of the urchins in a very short period of time. The kelp that recruits to those areas that urchins have been cleared from are all going to be about the same age. So you end up with a kelp forest composed of individuals that are one or two years in difference with each other. So pterygophora, for example, can live 15 to 20 years. And what that means is if you have a kelp forest that is composed of individuals that are all the same age, they're all going to age out at once, and the whole kelp forest will collapse at once. And you end up with these windrows of kelp on the beach. And just so you know, that kelp gets put to good use. This is actually an eagle's nest right here that is made largely from these beach cast pterygophorus stipes. So that little urchin response that we just looked at may prove to be quite important, because if you think about it, what happened there was the urchins were removed from the site over a much longer period of time, which may enable kelp forests to be composed of a much wider age range of plants, which might provide more stability. So I want to point out there are some other changes that are going on, and these are quite recent changes that we've only really just begun to notice. So pterygophora is a long-life plant. It lives about 20 years. That's work done by Rob de Vrede at the University of British Columbia. And it lives all the way down to Southern California, and it does okay down there, but it really much prefers to be in cooler waters up here to the north. This is a plant that reproduces during the winter months. So what it's essentially doing is reproducing at times when waves are ripping up kelp forests, and hopefully the spores that are produced from pterygophora can settle into the holes that are created by these winter storms. However, this is a plant that doesn't do super well when it gets really, really warm. So if water temperatures go above about 18 degrees Celsius, this plant can't make it. So there's a new kid on the block, though. It's not a new kid on the block. It's a plant that's been around for a long time, but it's been quite rare. And this is a plant called Icinia Arborea, and it's a stipitate kelp. But like pterygophora, it's quite long-lived. It lives greater than 13 years, about 13 years. And unlike pterygophora, it likes warm water. It occurs all the way down the Pacific coast of Baja California, but it does just fine up here in cool water as well. This is a plant that is reproductive in summer months. And of course, during the summer, water is fairly still, plants are getting torn up. So in the past, Icinia has always had quite a hard time getting a foothold. However, this is a plant that does just fine when temperatures get warm. Most of our other perennial species start to suffer when temperatures get over about 14 degrees Celsius, 14 to 15 degrees Celsius. They start to get limited by nitrogen. They start to get a little bit unhealthy. And so what appears to be happening is that Icinia Arborea, which has never been able to do well because there's never any holes in the kelp canopy in the summer, has been able to take advantage of these collapsing pterogophora forests and to take advantage of pterogophora that is being stressed out by what are increasingly warm water temperatures. And this is Gull Island. I want to take you back to Gull Island yet again. And this is, we're now density per meter squared across all of those 5 swaths again. So this is 1987 through to 2021. And there's that crash in pterogophora as it aged out. And here it is sort of trying to compete its way through that Desmorastia. It increases and crashes, it increases a bit, and it's doing okay. But here in the blue down the bottom is Icinia, which was pretty much absent from this site prior to 2005. And it has been steadily increasing in abundance. And in 2020, it became the most abundant stipitate kelp at Gull Island. And this is a pattern that we're seeing throughout the area I work in, the Checklisset Bay and Cayucet Sound. And this is a bit of a confusing graph, but there are 40 sites that we sample every 10 to 7 years. And we go down and just count everything in 30 quadrats. And so you have 2 measures here. One is the density, the average density of Icinia, and this is the number of sites with Icinia. And in 1988, when I started this work, there was no Icinia at any of those 40 sites. By 1994, 7 of the sites had Icinia at them, and they had grown to a density of about 1.15 per meter squared, not very many. By 2007, about a quarter of those sites had Icinia at them. The abundance had increased a little bit. But what I want you to notice is by 2019, almost half of the sites we sample now have Icinia at them. So this is a pattern that is widespread across the entire area I work in. And it's probably a product of increasing water temperature. It's a native species. It's a species that is resilient to warm water, so perhaps this is a good thing. But I just want to point out as I end up that Icinia is probably going to continue to thrive when waters are warm, and there's every indication that water temperatures are going to continue to warm. But there have been lots of changes, and this will be my second to last slide. I can't talk about kelp without talking about this guy. And this is the stellar sea cow, which was the largest of the Sirenia or dugongs. It was an animal that was 10 meters in length. It weighed about 10 metric tons. It was a species that stumbled around the shallow subtidal, eating what all dugongs and manatees eat, vegetation. This guy was a kelp specialist. And there's a lot of theories as to why it declined. One theory is that with the decline of sea otters, with the arrival of human beings, that kelp became less abundant, and this large Sirenian simply couldn't find enough kelp to eat. Whatever the case, it made its last stand on the Commander Islands, and it was described by George Wilhelm Steller in 1745 when he was shipwrecked on the Commander Islands in Bering's expedition. He described the stellar sea cow, measured it. This is a drawing that Steniger made of the stellar sea cow. By 1748, the species was extinct because it was easy to catch and it tasted like veal, and it provided ships heading across the Pacific Rim in hunt of sea otters. And so this is the species that's gone that ate the kelp that the sea otters made. And with that, that's all I have to say. I would like to acknowledge, I keep saying I and me, and of course it's not, it's a cast of thousands that have done, well, hundreds that have done this work over the years. I've been very blessed to gain a number of very close friends through this. I think I saw Leah and David somewhere on that list. Leah Seville has been out almost every one of these 35 years. Without her, I certainly couldn't have done most of this. The Friends of the Ecological Reserves sponsored the work from very early on, and I remain extremely grateful to them. And I've listed off a number of funding agencies. This work was done out of Cayucit, a lot of it, which is a small community on the northwest coast of Vancouver Island. And I'm very grateful to the Chaklasat and Cayucit First Nations who have been very welcoming. So thank you very much. That's all I have to say. And I can get out of here and become a face of many faces again.
Great. Thank you very much, Jane. I'll read a few of the questions that appeared on chat, and maybe you could answer them for us. I'll just go back to the beginning. One question from Jennifer Sutton. If the sea urchins need to eat so much and they decimate the urchins, what is happening to their other food sources?
Oh, so sea otters eat a wide variety of invertebrates. Sorry, I should have made that clearer with that first slide. So urchins are big, they're abundant, they're easy to find. So sea otters move in and they eat what is big and obvious first. Over time, what happens is the prey gets much smaller, but all of that kelp, of course, is increasing growth rates and fueling productivity. So even though the very large, big prey have gone, there's probably an even greater biomass of smaller individuals of the same species. So the prey that otters are eating gets smaller in time. They probably have to spend more time foraging, but they still do fine.
They eat abalone, don't they?
They eat abalone. And one of the things I quite often talk about is how we sort of came along into this system when sea otters were gone, and our impression of what the ecosystem looks like is all of these very, very large, very, very abundant species. But in actual fact, that's an artifact of having removed, probably, certainly for subtidal organisms, what was their major predator. And if we could turn the clocks back in time, sea otters have been around 600,000 years. So most of these species, abalone and urchins alike, probably co-evolved with sea otters, and they probably were much smaller and probably had life history characteristics that suited them to living, in cryptic crevices and places like that where sea otters couldn't get at them. But we sort of have this idea that abalone should all be 100 millimeters and urchins should all be 100 millimeters and so on. Probably wasn't the case.
Right. Another question from John Neville. Do Biggs killer whales eat sea otters?
Oh boy, that's a hotly debated topic. So the man I did my PhD with, Jim Estes, published a series of very influential and I would say important papers looking at a massive decline of sea otters in the Aleutian Islands, going from 200,000 down to a few thousand animals in a very, very short period of time. And he proposed that this decline in otters was due to predation by killer whales, and pointed out that because sea otters don't have any blubber, a killer whale has to eat a lot of sea otters to make a living. So he proposed that killer whales ate sea otters, and he went on to propose that sea otters were definitely not their preferred food. They would rather eat fatty, blubber-laden marine mammals like seals and sea lions. And he proposed what he called sort of a fishing down the food web hypothesis, where he said that illegal whaling reduced the whales. And so the killer whales had to go from whales down to seals and sea lions down to sea otters. And there's a lot of people accept that story. And I personally believe that sea otters probably occasionally get taken by sea otters, but by killer whales. But my husband works on killer whales. He refers to sea otters as dental floss, because there's really not very much in an otter for a killer whale. So on our coast, we haven't seen anything remotely like this. I have watched killer whales hunting sea otters 3 times now. In every single case, they were juvenile big killer whales that were practicing hunting. And the only outcome for the hunt were 3 very neurotic sea otters, because they weren't killed. They were just harassed and threatened. So maybe killer whales eat sea otters in Alaska. There are a few other hypotheses out there. They don't seem to eat a lot of sea otters in British Columbia. There was recently enormous stir caused by a dead killer whale washing up in the Commander Islands, and it had 6 sea otters in its stomach. They were intact. They hadn't even peeled the fur off of them, which is incredibly unusual. So I don't think it was a very healthy or happy killer whale. So that's a long answer to a simple question, but it's not a simple question.
Another question here from Jeff Krause is basically about one of your techniques. The density of algae, is it basically the number of stems per unit area that you're looking at?
Okay, it depends what we're counting. Excellent question. I know people want to hear about sea otters, not seaweed, so I try not to bore them too much. So when we're counting something like the giant kelp Macrocystus, we count the number, I would report the number of stems, but we also try and figure out what a plant represents, but it's the number of stipes of Macrocystus. And then all of the other ones are just single stems anyway. So they are by nature a single stem. If you're interested in kelp, the other thing we do is anything under about 15 centimeters gets called a juvenile kelp, because you can't always tell what species it's going to be. So they're sort of lumped in as well. I hope that answers the question.
Here's another one from Jennifer Sutton. How are the Indigenous people dealing with the lack of sea urchins?
That's another great question. There's the First Nations are not happy about the absence of tutsup, or where I work, urchins, and other shellfish resources. And I'll add to the list of unhappy people, lots of commercial fishermen that harvest geoduck and urchins. And it's an interaction that my colleague Anne Solomon and another colleague Ian McKechnie at the University of Victoria, Anne's at Simon Fraser University, have been looking at. And I suspect that it's going to turn out to be historically probably a very complex relationship. So where I work in Chuckleset Bay, there's probably, I don't know, 1,200 sea otters. And I suspect if you were to turn the clock back 500 years, when there were probably 10,000 human beings living in Chuckleset Bay, sea otters and human beings would have eaten the same resources. So there were probably fewer sea otters, just because they would have been competing with human beings. And of course, humans would have hunted sea otters to some degree. Now, because they're mostly hunting them for furs, I am of the suspicion that hunting probably didn't play a huge role in directly limiting numbers. And this is just my opinion. It's just based on thought processes. But we know from looking at other animals, wolves and elk in Yellowstone Park, for example, that sometimes animals will respond with fear and change their distribution or habits. So it may be that sea otters were distributed differently back in the day when there were tens of thousands of people wandering beaches and harvesting resources, and otters that were avoiding the presence of humans. And then a graduate student at the University of Victoria in Chris Derriman's lab, Erin Rechsteiner, has just finished her PhD. And she did a lovely chapter in her PhD dissertation that suggested that things like clam gardens may have played a very valuable role in actually reducing the ability of sea otters to access clams just by raising the clams up and reducing the length of time that sea otters could forage on them in the intertidal. So that's, again, a long answer. There are lots of First Nation groups engaged in wildlife viewing right now. So those people love sea otters. The people that want to eat toots up and clams, not so happy. So it's a mixed bag, I think.
Right. Yeah. There's another question from John Harper. Are you comfortable that your sites are representative of larger spatial areas?
Absolutely not.
Checklisset Bay?
Absolutely not. Those permanent sites are just that. They're permanently marked sites. And I didn't talk too much about my sampling design and approach, but those 40 sites that I alluded to at the end there with showing the Icinia increasing over a much broader area, those were randomly chosen sites, and the quadrats that we sample at each of those sites are random. So it is those 40 sites that I would say are representative of what is going on across Checklisset Bay and Cayucot Sound. What the permanent sites let me do is they let me know for sure that I'm documenting changes over time and not changes in space. So those two sampling methods, and I really didn't talk about the quadrats that much, are aimed at very different approaches. Hopefully that answers that, Don.
Okay. Yeah, here's another one from Mary Morris. Can you tell from sea otter DNA if the animals on our coast today likely all descended from those few 40 some individuals that were reintroduced?
So there are no unusual alleles. There are no markers that are unique to our sea otters that don't occur in Alaska. So all of our sea otters came from Alaska. Are all of our sea otters descended from the 89 sea otters that were introduced? Probably not, because at the same time sea otters were being introduced to Checklisset Bay, they were also being introduced to Washington State. And otters tend to do a funny thing when you translocate them. They try and swim home, and home was north. And so sea otters probably swam from Washington State into British Columbia. They introduced sea otters to Oregon. That was a failed translocation, but I suspect those otters probably swam to Washington State. And I suspect some of the sea otters that got introduced to the Bunsbys swam north and ended up in places like the Goose Group Islands on the Central Coast. So all of the animals on the BC coast are descendants of Alaskan otters that were translocated, but maybe not from the actual 89 otters introduced to British Columbia. There's no evidence for a remnant population.
Great. Question from Don Kramer. Do you know how long urchins avoid areas with tests of predated urchins? It seems surprising that the chemical signal would remain for weeks or longer.
It is amazing. We did that experiment for 10 days, and the quadrats, the ones with urchin tests in them were still at much lower densities than the other quadrats. In the summer, a spore can be released from something like a bull kelp. It settles down and it produces gametophytes, so the male and female version of the reproductive cycle, and you can get a little tiny sporophyte within 14 days. So although I mean, I was astounded when we did that, and Leah's here somewhere. Leah helped do this with me, and I am so glad she did, because I don't think anyone could believe how strong that response was. And there was someone else there saw it, but anyway, so it certainly is possible, but it's the only mechanism that I can come up with to explain that pattern.
Right. And final question here from Jacques Sirois. Do we know how abundant sea otters were historically in the Salish Sea?
So there's no historical record of sea otters in the Salish Sea. To my knowledge, the nearest otters found in First Nations midden sites are from the Souk Basin. And so this Aaron Rechsteiner, who did the clam garden study, compared clam gardens in the Strait of Georgia to clam gardens in the Broughton Archipelago, her idea being that in the Broughton Archipelago, they deterred otters. And she could find, working with archaeologists, absolutely no evidence that sea otters were in the Strait of Georgia in middens back, I think, about 3,500 to 5,000 years. So there's no reason why otters couldn't be here. I mean, there are a few otters in the Strait of Georgia, but I suspect there were an awful lot of people living in the Strait of Georgia, and they probably were hunted. So, but there's no evidence they were abundant.
Right. I was just wondering myself, do you have any sense of how much more they're going to be spreading south and around? We occasionally get sightings off Race Rocks.
Yeah, the predicting spread rates is one way to be wrong for sure, I think. Excuse me, they have a very erratic way of spreading. The males spread and then the females fill in behind, so you sort of get these big skips. And it really depends on things like habitat. So places like Long Beach, it's not very good habitat, too sandy, too exposed. So they'll probably, you know, they jump over that. So no, I have no predictions. They're not really in Barkley Sound yet. And I think I would have expected them to have moved into Barkley Sound by now, but there's, you know, a few scattered individuals. So I don't know.
Yeah. Okay, so are there any more questions? If you'd like to unmute yourself, if you have a question. So, hearing none, I will now thank you, Jane Watson, for an excellent talk. You're much wiser now about kelp and the effects of sea otters. Yeah. So I'd like to thank you very much for your time, and it was an excellent presentation.
Thank you very much. Thank you to all of you for putting up with kelp.
So I'll send you the maybe the chat afterwards, just so you can see. There's a lot of thank yous coming up now too.
Oh, thank you. Thank you, guys are very gracious.
Yeah. Okay, so I'll with that, I'll say thank you and sign off.
Bye-bye all.
Bye.